Hello, my name is Lindsay Shuck. Thank you for joining today's Family Office Network's webinar series featuring Akamara Therapeutics. I would like to introduce our speaker today, Susan Graff. Susan is an accomplished global biopharma executive with 25 years of strong industry experience in both large pharma and small biotech. Prior to Akamara, Susan was Chief Business Officer and Principal Financial Officer at Epizem, Vice President, Corporate Development and Strategy for NPS Pharma, and led the company's sales. Susan also spent nearly 18 years at Roche. She was recognized as a Healthcare Businesswoman's Associate Rising Star in 2011 and has been named to Purdue University's 40 Under 40 list. At the end of today's presentation, we will have time for Q&A. Please use your questions drop-down option to send over your questions so Susan can answer them accordingly.
Thank you, Lindsay, and good afternoon, everyone. It's my great pleasure to introduce you to Akamara Therapeutics, which will be the company known for reimagining immunotherapy, both in cancer as well as rare disease. We intend to deliver on this vision by really focusing on inventive B-cell therapeutics and seeking to change the way people live. We believe that focusing on novel B-cell therapeutics and the insights we've gained with our lead program, AT1965, will really allow us to build a deep pipeline beyond our lead, not only for cancer immunotherapy, but also for the treatment of other B-cell driven rare diseases. Our lead program is in a late preclinical development stage. We have recently had a positive pre-IND interaction with the FDA and reached alignment on our IND submission, and that will happen toward the fall of 2020. And our ambition is to have our first in human clinical study open by end of this year, and that will be in cancer patients that have solid tumors. The company has largely been backed by Accel, which is the large tech venture group that is building a healthcare vertical to date. I also have a number of family offices and high net worth individuals sitting on my cap table. We are underway with our financing activities to continue to fund the growth of the company and drive AT1965 into the clinic and through our first in human study. We've executed a Series A financing led by Accel at a pre-money valuation of $18 million. We may explore topping off that round if there is any immediate interest based on some other investors who've recently approached us. But the main financing plan for 2020 is to initiate a Series B raise to fully fund the first in human study of 100 cancer patients that I'll tell you more about shortly. In terms of the team that is behind this company, Lindsay gave you a background on me, so I'll just introduce the rest of my team. I have Dr. Sergio Santillana as my Chief Medical Officer. Sergio has a number of different oncology approvals to his name, notably through his time at large companies like GSK, as well as small biotech like Ariad. He served as CMO of 2 other biotechs, and I'm very fortunate to have him leading the design and implementation of our first in human study for AT1965. I'm also joined by Chandrasekhar Natarajan. He is my head of research and translational medicine. Nat served at Sanofi in a variety of different senior roles, ultimately becoming a known ADME and PK expert. He has since retired from Sanofi, is on adjunct faculty at Tufts, and spends 4 days a week with me, really at the moment focused on our translational and biomarker approach as we advance into the clinic. I have Dr. Jim McArdle as my head of CMC and quality. Jim also has large pharma and small biotech experience on the CMC and quality fronts. He served on key working groups of the IHC and has several IND filings to his name. I'm also joined by my 2 founders at the bottom of this page, Dr. Monadeepa Roy, who is a cancer biologist trained at Harvard. She has kept an operating role with Akamara and serves as my Vice President of Corporate Development and Operations. And my other founder, Dr. Shiladitya Sengupta, who is faculty at Harvard Medical School, he remains a board member. This is the team that we have to drive our lead program into the clinic. We absolutely have the experience to do that and to capitalize on all of the value creation opportunities that are in front of us. We supplement the internal team with an esteemed panel of scientific advisors. We have the spectrum covered again from cancer biology with Dr. Bruce Zetter to B-cell expertise with Dr. Ricardo Dalla Favera. We have Dr. Shireen Loy from Australia, who is a translational expert and very specifically focuses on T infiltrating lymphocytes and now is very interested in B infiltrating lymphocytes. We also have Dr. Aditya Bardia, who has served as a principal investigator on a number of different studies. And we're benefiting from not only the preclinical aspects of our program, but as we move into the clinic from this expertise. Let me tell you a little bit more about the space before I get in specifically to our lead program. We are in an era where checkpoint immunotherapies such as Keytruda and Opdivo have really made a meaningful difference for cancer patients. But we need new angles to explore the cancer immunotherapy landscape from, because we need to provide some additional benefit. And B cells is one of the ways that the cancer immunotherapy field is going to evolve. What you see on this page is a series of 4 articles published in Nature from January 2020. This is just a statement to show all of the scientific literature that we're seeing that shows B cells are the next frontier in cancer immunotherapy. The Nature series, which I just showed you, is only one piece of the literature that's out there. There really is this wave of persuasive science that continues to show B cell activation can play a role in ultimate survival of cancer patients. And this is an important therapeutic approach that should be explored in human cancer. You can see in the image here, B cells and T cells, they talk to each other. There's a lot of crosstalk here. And the concept is simple. T cell targets, we know which of those work to date. We've not yet explored the B cell activation angle, and B cells have the unique property of truly being able to elicit a very durable memory in response to something like an antigen, a tumor antigen. And we truly feel that when we think about what cancer immunotherapy will look like in the future, that B cell activation approaches, with Akamara's 1965 really leading the way here, will be what allows us to achieve this promise of adaptive immunity in cancer and truly allow the body's immune system to be unleashed against some of these solid tumors. I had mentioned the checkpoints and the benefit they were able to provide. On this slide, if you look at the green line, that is really effectively where we are in terms of the proportion of patients that are able to achieve this, what we call the long survival tail from current cancer immunotherapy. Akamara is really looking to shift the line, up at least to the pink line, perhaps higher ultimately when we move into combination. But with our B cell approach, we truly do believe it's paradigm changing, much like the checkpoint inhibitors were when they first came into the cancer immunotherapy scene. We know that we can also track B cell activation in the clinic. So what you're looking at here is a estrogen receptive negative breast cancer population published in literature where they're tracking a mark called IGKC. This is immunoglobulin kappa chain. It's a mark uniquely expressed on activated B cells. And here you can clearly see from the Kaplan-Meier on the left, the intense separation of survival curves for patients who had high IGKC expression, which is a surrogate for high B cell activation, versus those that did not. This again encourages us that B cell activation can correlate with outcomes and survival, and can be measured and tracked in the clinic as a parameter far beyond just the objective tumor response that we will also be looking for. In terms of AT1965 as a lead program, again, this is paradigm changing, and we are leading the way to the clinic here. Our mechanism of action, we're not able to disclose today without a CDA because we're following up on some provisional IP. What I can share is that this is a small molecule approach. It is first in class, and it is a molecule that is interrogating a novel cancer target that has not previously been in the clinic, nor are we seeing intense competition for this target right now. We have a composition of matter patent issued on AT1965 that goes through 2033 without extension, so nice IP coverage as well. I'm now going to walk you through our key in vivo animal data that got us so excited about AT1965 as our lead program. What you see on this slide are a number of different syngeneic mouse models looking at 3 different solid tumors. On the far left, we're using the 4T1 cell line as reflective of a triple negative breast cancer model. And you can clearly see in the blue line the striking tumor regression that we see when we treat these animals with AT1965. This is a very difficult to treat cancer, and this was what really put us on the path of the next sets of experiments that I'm going to show you to truly understand why that regression was happening. If we look in the middle panel for melanoma and on the far right for Lewis lung, again, AT1965 is reflected in the blue line, and you can see in all cases that there is tumor growth inhibition relative to control. Taken together, this gives us confidence that we should expect anti-tumor activity across multiple different solid tumors. Following up on the triple negative breast regression data that we just showed, this was our first key experiment, which we call our immune memory experiment. Again, syngeneic mice. We used the 4T1 tumors. If you look at the top row of mice, this is our experimental row. We implanted those 4T1 tumors. We treated the mice with AT1965 and saw the tumor regression as I just showed on the previous slide. We then waited 25 days. Challenge those mice with 4T1 tumors, and what you see are the striking results on the top right in the blue line. Upon re-challenge, the tumors did not engraft. We saw the initial tumor regression, and then upon re-challenge, we saw what we believe is an immune memory kick in and prevent that tumor from engrafting. That's in stark contrast to the 2 control groups we looked at in non-tumor bearing animals. The first control was treated with our compound, AT1965. The second was treated with saline. We waited and challenged both of those control groups at the same time point as the experimental group with 4T1 tumors. And you can see the results on the bottom right. Both control groups grew as you would expect. And in stark contrast, AT1965 had induced an immune memory that caused tumor regression and continued tumor suppression. Following this up, we looked at the 4T1 model in a variety of different immune contexts. On the far left, you see immunocompetent wild-type mice. And what you see here again is a tumor growth, regression, and inhibition in this model in the purple line. When you knock out the entire immune system, looking at the middle panel in our SCID mice, you see AT1965 loses all of its activity. This indicates that a functioning immune system is necessary for the compound to have activity, and we're not seeing direct cytotoxic activity from the compound. We further looked at what would happen if we knocked out just the B cells, and this is in the far right panel, using a JH knockout model. And here you can see again in the purple line that AT1965 really loses the vast majority of its activity when the B cells are knocked out. With any immune system, there's going to be cell crosstalk. I've already showed you that. But we believe here that the B cells are the key orchestrator or the quarterback of the immune response that we're seeing. And after that last JH knockout model, we followed that set of experiments up with this model to kind of truly confirm that B cells are the driving immune cell type here. This is called our adoptive B cell transfer. What we did looking at the mouse schematic is implanted an experimental group of mice with 4T1 tumors, treated with AT-1965, and again saw the tumor regression as we've seen in the past. We then harvested the spleens of these regressed animals, isolated the B cells, and we call these now our primed B cells. The reason we call them primed is because they have seen both AT-1965 and they've seen the tumor. We then infuse those primed B cells through what we call our adoptive B cell transfer into a group of non-tumor bearing mice. We then challenge those mice with 4T1 tumors. And what you can see again are the results in the line graph on the right. You can see in the purple line AT-1965 treated B cells. These are our primed B cells. You see tumor growth inhibition just from the B cells that have seen the tumor and seen AT-1965 previously. Again, AT-1965 was not directly administered to these animals. This is a striking result for us, again, relying on the immune memory that was previously generated. Again, that's in stark contrast with both a saline control, which you can see in the green line, and with control naive B cells as well. So you cannot induce this effect by just injecting naive B cells. It has to be a primed immune response. These are the key animal experiments that have led us to advance AT-1965 to the point of an IND. Let me just share with you exactly where we are on this path for 2020. We have completed our GLP toxicology study in both rat and dog. We are in final reporting phase for those studies. We have completed a successful pre-IND interaction with the FDA, as I've already mentioned, and that interaction really confirmed that the comments from FDA are largely around the first-in-human study protocol, and that we have a clear path to an IND this fall. We have produced our GMP drug substance, and our GMP drug product is slated for production late summer. We have engaged our clinical research organization who will run our first-in-human study, and this is also the same organization who is helping us author our IND modules. Again, targeting an IND for late summer, early fall of 2020, and our target is to have our first-in-human study open for enrollment by December of 2020. Let me transition now and tell you a little bit more about this first-in-human study. We are approaching the study from what we call clinically enriched patient population. We did not want to do an all-comer oncology study here, and we actually asked FDA this very question, and FDA did agree with this clinically enriched approach. The way we're clinically enriching is to really look at, first and foremost on the left, the tumor types that we have in-house data on: triple-negative breast cancer, non-small cell lung, melanoma, and colorectal carcinoma. All of these models, AT-1965 has demonstrated activity, and in all of those tumor types, B cells also have a positive correlation with outcomes and survival. We're then supplementing that study population with the tumors that you see listed on the right. This is a list of tumors that was selected largely based on the human literature that I've been talking to you about that shows B cells play a role in the outcomes of these tumor types. You also see a number of tumor types listed here that, should we see AT-1965 activity, could serve as an expedited regulatory path. If you think about something like cholangiocarcinoma, if we were to see a signal there, you could potentially imagine an approval on something like an objective response rate surrogate endpoint with supportive duration of response data. So this is how we've chosen our first-in-human study population. And we are intending to take that study population into a classic dose escalation followed by dose expansion study. The dose escalation phase, we expect to be roughly 40 patients. That will take us roughly 12 months to complete. And then our target right now is to have roughly 3 different solid tumor cohorts in the expansion phase of slightly more than 20 patients each. We'll obviously inform what those expansion cohorts are as we learn more about the clinical activity through the dose escalation here. Importantly, though, we want this study to be of robust size and be this 100-patient size so that we really have every chance to detect an early signal of efficacy far beyond learning about the safety and the profile from a PK and PD standpoint of the compound. I just want to also mention here, in terms of the exploratory biomarker program that I previously mentioned, that will be from first patient, and we will be looking to measure things like IGKC to measure that level of B cell activation that we may see. We'll also be looking at a variety of different B cell targets just to track cell movement. With that, I will wrap by talking to you about the use of proceeds that we are planning from the ongoing financing activities. Importantly, we're in that critical IND preparation and imminent submission stage. So we're using funds for that right now. We are looking to fund the full 100-patient study with the robust PD and biomarker program alongside of it, which I just showed you. We are currently staffed as a preclinical discovery organization. We obviously need to evolve into a development stage biotech here. And while we haven't spent much time today, we are also earmarking some of these funds to continue to really expand our pipeline and unlock that potential of B cell therapeutics. We have another IP platform within the company. It's a linker technology called Magnet. That could allow us to create antibody drug conjugates, both for our own portfolio, but also for potential strategic collaborations. It could also allow us to create bispecific type scaffolds without having an actual antibody in the middle. So powerful technology that we would like to invest and advance further. We also have some other early discovery cancer programs around B cell activation that we would like to move forward. With that, I will wrap by just sharing that we are very excited about the potential of Akamara to really reimagine immunotherapy here. We're excited about the prospect of having our lead molecule, AT1965, in a human cancer trial by end of this year, and really seeing the clinical benefit that an approach like this could provide in the next 24 months. The team is the team that can take this company forward, and I look forward to taking any questions that you have at this point.
Susan, thank you so much. Again, we do want to encourage everyone to send Susan questions as we have time to open the dialogue. There's a questions drop-down option for you to chat over any questions that you have as we jump into Q&A. Susan, we do have a question. What cancers are you specifically targeting?
So with a first-in-human study, FDA typically wants you to do what they call an advanced cancer study. That means any patient who has no other approved treatment options should be eligible to come into your study. I'm going to go back, we are very specifically narrowing that slightly to have a clinically enriched solid tumor population shown here on this slide. So you see triple negative breast, non-small cell lung, melanoma, colorectal. Those are all patient types that we will look to enroll, as we will ovarian, cholangiocarcinoma, soft tissue sarcoma, hepatocellular, head and neck, and cervical cancer as well. So this will be the first study population. And then depending on where we see signals of objective response, will define what our dose expansion populations are. And it will likely be 3 of these tumor types in the dose expansion phase. I hope that addresses the question.
Thank you so much. We have a question from Barry. When do they expect acquisition?
Sorry, Lindsay, expect what?
Acquisition?
Acquisition?
Yes.
So Barry, this is a company that, as you can imagine, based on cancer immunotherapy exits previously, we've got a couple value creation points likely in front of us in the 2021-2022 timeframe. We're raising the Series B later this year. In 2021, we'll squarely be in the clinic. I could envision either a crossover round into a public offering on the back of some human data, or I could also envision that a strategic who's deep in the cancer immunotherapy space could interrupt our plans with an unsolicited M&A offer. I would expect that we will need some clinical data on that. That does seem to be the trend if we also just look at the Gilead acquisition of 47, which is for a different type of target, not a B-cell target, but was cancer immunotherapy, and that deal just happened in March of 2020 for $4.9 billion. So I expect, again, Barry, once we have some clinical data, that there will be a number of different opportunities open to us.
Wonderful. And Barry is actually kind of following up, and he would like to know what the biggest hurdle that you see so far would be.
I am mindful of the fact that we are opening a clinical study as we, go forward into a potential another wave of COVID. So we are just being very deliberate with our CRO partner who runs a ton of oncology studies to take the learnings that they've actually had in this first experience to make sure that the clinical study, beyond the patient population that you see on this slide, but that the clinical study is most fit for purpose so that we're able to move it quickly and as we would expect. I don't know that I would consider it a hurdle necessarily, but we are being thoughtful about that, and that is one of the things on my mind as we move this program forward.
Perfect, thank you. Are there other companies pursuing B-cell activating therapies in cancer that you know of?
So there are other companies pursuing B-cell approaches, but we've done a pretty extensive competitive analysis, and we're not finding other companies that at the moment have a clinic-ready B-cell activation program for solid tumors. A lot of B-cell work in cancer has previously been done in hematologic malignancies, like non-Hodgkin's lymphoma, because in those diseases, the B cells themselves are actually the malignant cells. So they're looking to knock out B cells, not activate them. But learning from that, you've been able to take B cell therapies like that into autoimmune diseases. So I would say if you went out and did a search, you will find B cell companies. What you won't find are B cell companies applying an activation approach in solid tumor cancer immunotherapy. So I expect, because of the Nature series and all of this other literature, that the space will get very crowded, but I think we are fortunate to have a very nice head start versus any others who would be going after this type of approach.
Great, thank you so much, Susan. We have a question from Richard, and you may have touched on this a bit. How much money have you raised so far, and how much money are you looking to raise now?
So the Series A, which was led by Excel, was $16.5 million. That has all largely closed. Like I said, there is some intense investor interest right now. We are considering a small top up on that round, so there may be another million or two that comes into the Series A. We are looking to raise in the Series B in the order of magnitude $30 to $35 million. That would fully fund this first-in-human study that you see, and it would support the creation of Akamara as a development stage entity.
Perfect, thank you. Susan, how long do you expect to be in clinical before you start seeing clinical activity?
Let me just go to this slide. So the dose escalation, as I'm sure you all appreciate, you have to start at a sub-therapeutic dose to start exploring the safety profile when it's the first time you've put the molecule into humans. So we expect probably around that 9-month mark, so toward the end of the third quarter, after we're in humans, that we will start to be at what could be a human efficacious dose for the compound. So call it between 9 and 12 months is when we might first start to see that activity. Obviously, the final starting dose will be agreed with FDA at the time of IND submission, but that is our current estimate.
Perfect. Again, we do want to encourage everyone to submit their questions over via their questions drop-down option so Susan can go ahead and answer them. We do have a question. What are the merits of a B cell activation to cancer versus the T cell approaches? You can elaborate on that a little bit.
Sure. Thanks, Lindsay. So the T cell approaches are really a well-trod path. They are approved, as we've talked about. They're the PD-1 inhibitors, PD-L1 inhibitors out there, the Keytrudas, the Tecentrics, the Opdivos of the world. There are also things like CTLA-4 inhibitors. You also see cell therapy after T cell targets like CD19. You also see some antibody therapy there. It has just been a space that once clinical activity started to be demonstrated by the PD-1 inhibitors, and actually CTLA-4 in advance of that, became a very active cancer immunotherapy space. And B cells were very active, but again, it was frankly more on the autoimmune side or the hematologic malignancy side. There has not been a lot of application to these B cell approaches. But what I want you to think about is B cells are the way that your body ultimately fights foreign pathogens, fights a bacterial infection, fights a virus, actually produces antibodies to such pathogens. B cells are actually how immunizations for COVID, for example, will ultimately work because they are able to provide this durable, ultimately humoral memory that T cells, while there is definitely some memory associated with T cells, their role is not that long-term durable memory. That is the role of B cells. So we really are looking to change the equation here around how we can initiate an immune response against cancer by going after B cells and that long-term durable immune memory response that you saw so clearly in our preclinical models.
Thank you so much, Susan. We do have another question from Barry, and we sort of touched on it earlier, but he is asking how much money we're raising now and how much in the future.
So Barry, the 30 to 35 million in the Series B now, that really will get us through this first in human study. A regulatory path is always hard to predict in oncology because should we be fortunate to see significant clinical activity in our expansion cohorts, for example, we may be able to just expand this first in human study and ultimately file a regulatory NDA on such study. That's actually what Keytruda did when Merck brought that program forward. Their first filing was on an expanded phase 1 study. So I can't really comment, Barry, on how much into the future because it really will depend on what a regulatory strategy is after we see the activity from this first in human study.
And Barry, if you have any other questions, I'm sure Susan would be happy to touch base with you offline regarding the raise. We do have a question from Tom. If B cells can be cancer causers, have you seen the activated ones become a problem after they attack tumors?
We have not seen that. And in fact, the literature, that is really being explored there does not suggest that in human cancers that the B cell activation will be cancer causing in any way. The diffuse large B cell lymphomas, the non-Hodgkin's lymphomas that I was referring to, there is an actual fundamental defect in those B cells. We're not talking about that in the solid tumors that we're going after. Hopefully that helps clarify.
And Tom, if there's anything else that you'd like to add on to that question, go ahead and send it over. For now, we don't seem to have any other questions. I'm going to take a few seconds here to just see if anybody has anything else that we can answer. Susan, in the meantime, is there anything else that you would like to add before we close out today?
I would just welcome any of you to contact me offline. I'd love to, take you deeper into the science and into this story, but I'm very excited about this approach. This completely novel angle of B-cell activation is what brought me to Akamara, and I'm truly inspired by the ability that this compound could help patients. And that's what I'm looking to do, and that's what Akamara is looking to achieve in the near term. So thank you for your time.
Thank you so much. And on behalf of everyone at Family Office Networks, we thank Susan and Akamara for being with us today. And we will be following up with everyone directly after this webinar. So if there's any other questions, we can send over and set up a call with Susan as well. We hope you have a wonderful day, and we look forward to speaking with you soon. Thank you so much.
Bye-bye, everybody. Take care.
Bye, Susan. Thank you.
Thanks, Lindsay.